My name is Noyusna Shahirah binti Muhammad Isman. The topic we chosen for our presentation today is paper chromatography. Now, I'm going to begin our topic by providing a clear explanation about chromatography. What is chromatography? Chromatography is a technique used to separate and analyze the component of a mixture. It involves passing the mixture through a stationary material, like a column or plate, while a fluid or liquid or gas carries the component through. Different components move at different speed, allowing them to be separated and identified. There are 7 main types of chromatography, which is gas chromatography, liquid chromatography, thin layer chromatography or known as TLC, paper chromatography, affinity chromatography, ion exchange chromatography, size exclusion chromatography. Today, we are focusing on one specific type called paper chromatography. Paper chromatography is a technique used to separate and identify substance within a mixture. It works on the principle of differential partitioning between a stationary phase, which is paper, and a mobile phase, which is solvent. As the solvent moves up the paper, it carries the sample component along, which separate based on their affinity for the stationary and mobile phase. This separation allows us to analyze the composition of complex mixture.
Theory chromatography paper. The principle can be partition chromatography or absorption chromatography. Partition chromatography is utilized because the components are divided or spread among liquid phases. The two phases are water in the filter paper's pores and a mobile phase that flows through the paper. When the mobile phase moves, the mixture separates. The compound in the mixture separate due to variation in their affinity for stationary and mobile phase solvents as a result of the capillary action of paper pores. Absorption chromatography between solid and liquid phases, wherein the solid surface of the paper is the stationary phases and the liquid phase is the mobile phase. Application of chromatography paper. First, to investigate fermentation and ripening. Second, to determine the purity of medication. Third, to inspect cosmetic. Fourth, to detect adulterants. Fifth, to detect pollutant in beverage and meals. Sixth, to study the reaction mixture in biochemical laboratories. Lastly, to identify dope and narcotic in humans and animals.
Hi, my name is Ainu Mayravin Turasli and I'm going to explain the methodology of this experiment. For the first step is to draw a parallel line with 3 cm from the bottom using a pencil. It is advised to use a pencil instead of a pen because the pen ink is considered a dye. The next step is to mark 4 small x's along the line equally distanced and was labeled red, blue, green and yellow for each axis. For the next step, prepare the developing solvent using 2 grams of NaCl dissolved in 100 ml of distilled water. The 70 ml of developing solvent was poured into a 1000 ml beaker. Next, lay the chromatography paper and use a micro capillary tube to place a tiny dot of.
Now let's move on our results and calculations. Based on the experiment that we have done, this is the final results that has been obtained. As you can see, there are 4 different types of food dyes with their components shown on the paper. So, red food dyes produce one component which is red color. Blue food dyes produce one component which is blue color. Green food dyes produce 2 components which are yellow and blue color. And lastly, yellow food dyes produce one component which is yellow color. For the calculation, we have calculated the RF value of each food dyes. We use the formula which is Rf is equal to the distance of solid move divided by distance solvent front move, which we got about 12 cm. Thus, the results of each foot dice are carried out as shown above. So, based on the Rf value, red and yellow foot dice have a good value compared to the blue and green because the impurities are resolved from each other.
Assalamualaikum and hi everyone. My name is Hanis Batrisha binti Abdul Rahim. My student ID is 2022617386. Today I am going to explain the conclusion for experiment paper chromatography. The experiment using gravimetric analysis and paper chromatography successfully demonstrated the separation and identification of components within a mixture. Gravimetric analysis provided quantitative data on the mass of the separated substance, while the paper chromatography enabled the qualitative identification based on the distinct migration patterns of the compounds on the chromatographic paper. The gravimetric method yielded precise measures of the isolated compounds, confirming the effectiveness of the separation process. Paper chromatography further validated these findings by showing clear, distinct spots for each component, indicating successful separation. Overall, this experiment highlighted the complementary nature of gravimetric analysis and paper chromatography in both quantifying and identifying the constituents of the mixture. The result confirmed the reliability of these techniques in analytical chemistry, providing a robust approach for both qualitative and quantitative analysis. Future experiments could explore the application of these methods to more complex mixtures to further evaluate their efficacy.